Good evening, ladies and gentlemen. It's always a dubious thing to be the last speaker, especially if it's been a long day. And I don't know how many of the international faculty are aware of the vagaries of T20 cricket. But you know, the last batsman usually has a very important role. Either he scores all the runs and makes his team win, or he goofs up and his team actually loses. So we'll decide whether we can win or lose, but the clock ticking there appears to be an enemy in any case. I'm here to discuss a topic which is, especially after one televised episode of Amir Khan's Satyamev Jayate, has led to a lot of national debate. And the national debate has been something which has been happening globally over a long period of time about the use of original research molecules or generics in our treatment and day-to-day practice. While in school and college, I definitely had socialist leanings. And I still subscribe to a socialist point of view when we are looking at a community or a country as such. But for us physicians, each patient has critical importance. And for that patient, the choice of each medication is truly critical. We know that biologic drugs, the history of biologic drugs started with Banting and Best. And we actually had insulin, which was then made into a recombinant compound. We now have a lot of erythropoietin. We use a lot of abciximab, interferon, and a lot of newer monoclonal antibodies. The term biosimilar is very confusing because in different regions of the world, different terms are used to describe similar things. In the European Union, whether it is generics or similars, they are all considered biosimilars. In the United States, these are referred to as follow-on protein products. In Australia, they're again biosimilars. So a whole lot of names are there. Let's just look at two stellar definitions. The Food and Drug Administration defines biosimilar as a biologic product that is highly similar to a US licensed referenced biological product, notwithstanding minor differences in clinically inactive components for which there are no clinically meaningful differences between the biologic product and the reference product in terms of purity, safety, and efficacy. Whereas the European Medicines Agency defines it, a biosimilar is a medicinal product. It is a medicinal product that is similar to a biologic medicinal product that has already been approved and authorized. Basically, a generic is a small molecule drug whose active substance is shown by appropriate testing to have identical physical, chemical, preclinical, and clinical properties to an originator small molecule drug. Whereas, what is a biologic? A biologic is a drug produced from a living cell, tissue, or organism through a biologic process. These drugs imitate natural biologic substances in our bodies, such as hormones, enzymes, or antibodies. And these medicines, such as vaccines, blood products, biotherapeutics, are very important tools for the prevention and treatment of disease. But these biologic products have very large molecular weights, and they have very complex structures, and they can be called as large molecule drugs. You can have bio-betters or bio-superiors, which may be recent iterations similar to innovator biologics, but some change in structure of protein or process of manufacture with having improved safety, efficacy, or immunogenicity. You can have super biosimilars, which can be second or third generation drugs. And these are, a biopharmaceutical is a drug product containing biotechnology-derived proteins. And these are usually very large. The process itself is the product, and the safety and efficacy is highly dependent on the robustness and the monitoring of quality aspects. Why biosimilars? The entire global economy is going to have to pay a huge burden in treating people. And biosimilars are actually a hot new area. Though biotechnology-based drugs account only for a fifth or so of global drug sales, they are projected to grow at double-digit rates, especially with a lot of patent expirations which are coming. So if we compare generics, what are generics? Generics are small molecules, so you can copy it very well. They are identical copies. Biosimilars are similar but not identical because they are large molecule. The generics need an exact composition, which is a chemical composition. You can have a carboxyl chain or whatever, whereas a biosimilar need research as a molecular structure may not be known and can be different and needs a good biotechnological product. Of course, both these will cost less than the original. What about the legal framework? In the European Union, there was an early introduction of a legal framework as a one-stop shop since 2003. And a lot of biosimilars, about 14 biosimilars have been approved in the European Union. The US legislation was more lax in comparison and it was more complicated than in the European Union. It was only in 2010, on March 23rd, 2010, that Barack Obama signed the Biosimilars Act, permitting these biosimilars to be introduced in competition and innovation. And in some European markets, healthcare reimbursements are based only on generic model and are too low to cover the cost of even the production of biosimilars. Why are these different? Complex structure, high molecular weight, three-dimensional structure, complex manufacturing process, living organism, very difficult to characterize, depend on a biologic activity, some immunogenicity. So we must understand that biosimilars are not the same as generic biopharmaceuticals. How are they made? A biotechnological process, develop a host cell, cell bank, protein production, some purification, analysis, formulation, storage, handling, and they can be excipients which are introduced. There is a huge global market. Erythropoietins, insulin, interferon, colony stimulating factors, growth hormones, all these are used by biosimilars. These are unstable and large molecules. The structure is held together by weak, non-covalent forces. They are easily destroyed even under relatively mild storage or handling condition. And because of this, it is very difficult to demonstrate bioequivalence. There is a huge rise in the amount of biosimilar market globally, and the debate between the innovator company and the generic companies on whether a biosimilar would have had the same result have been going on for more than a decade. Is this Satyamev Jayate? Is it a question of science or is it politics? They are not similar. They are not identical. They are similar. There is need of regulatory approval. There is no patent protection, and there is a need for assessing safety and efficacy. We need to understand that we have to define them carefully. We have to understand that this debate started from the time of understanding different molecules. Look at aspirin. It has only 21 atoms. It's a small molecule. It is like a bike, which is easy to replicate. But look at a human growth hormone or an antibody. It's a huge molecule. It's like a jet, which is very difficult to attribute. There are a lot of biosimilars which are actually approved, but we must have a patient safety and pharmacovigilance by which we can differentiate between the reference product and you can understand the interchangeability. The two biosimilars have different origins. They may have similar therapeutic effect, but may have different side effects and toxicology. Hence, they require thorough testing. And if you look at a meta-analysis of even use of generics, you will understand that although this was in JAMA in 2008, and the meta-analysis said whereas evidence does not support the notion that brand name drugs used in cardiovascular medicine are superior to generics, a substantial number of editorials counsel against the interchangeability of generic drugs. What is the difference? A generic can be something which is identical. It's like a mirror image. Whereas biosimilar is like a twin. It is similar but not the same. So, and you look at some of the issues which are created. Erythropoietin, a classic example. Erythropoietin was used by Johnson & Johnson and which was marketed all over the world. And this erythropoietin, how we identified different erythropoietins was to look at isoelectric focusing and see that the control was not the same as the biological products. Because the process itself is part of the product. Because it can have a minor change in process, can have an impact on potency, selectivity, pharmacokinetics, and immunogenicity. Let us look at the process. In a small molecule, the structure defines the product. Whereas in a biological molecule, it is the process which defines the product. And that is the difference. What happened in Eprex? In Eprex, the erythropoietin which was being marketed globally did not have HSAA, which is human serum albumin. And it was not understood why the erythropoietin, which was being marketed outside the US, was causing pure red cell aplasia. It took Johnson & Johnson 10 years to try and understand this because of an excipient. Look at Marvel insulins. Marvel insulins were an Indian insulin which was produced. And when we looked at all the data, we looked at a lot of insulin experience, it could never reach the level of being accepted in terms of quality or in terms of clinical aspects. Let us look at low molecular weight heparins, something close to our heart. We understand that low molecular weight heparins are actually biological products obtained from animal heparin by depolymerization, and no generic applications can be equivalent or are accepted. And a certificate of suitability is not enough to replace scientific data, because there has to be clinical profile, pharmacological activity, and chemical structure, which actually relates to a manufacturing process. Because when we manufacture these products, even for enoxaparin, we have to look at cleavage point in terms of fingerprints. And when we look at these fingerprints, we also look at some oligosaccharides containing classical process-dependent antithrombin 3 binding affinity site. And when we realize it, only about 70% of the molecule has yet been characterized, 30% of the molecule is actually unknown. So there are technological limitations in terms of oligosaccharides. The product cannot be completely characterized, and specific monograph fingerprints are required. So how can they be the same? There are differences, and the differences exist between enoxaparin in different brands which were used even in terms of molecular weight. There are differences in terms of anti-2A and anti-10A efficacy. So as a consequence, low molecular weight heparins are biological compounds which are not fully characterized. Process determines the product. Immunogenicity is an important safety issue. There need to be clinical trials and pharmacovigilance which help guard against immunogenicity. And biosimilars must provide relevant quality preclinical and clinical data to understand this. Unfortunately, India has a biological guideline, but it is not retrospective. So we must understand that similar is similar, but not the same. What about India? In 80s and 90s, many biological products were approved by state licensing authority. From 1st January 2009, we have a regulation, we have a National Biotech Regulatory Authority, but it is not retrospective. We need at least 2 double-blind trials. We need demonstrate non-inferiority and clinical, but what we need is robust pharmacovigilance. Because we have to understand that we must be able to trace, label, and have a complete process in place to see any new signal. And there's a great opportunity for Indian pharmaceuticals which are actually ruling the world. And in India, we need to study this issue, need to organize expert body, need to identify super experts, need to listen to it from the mouth of horses, need to strengthen the pharmacovigilance plan. Just a few studies, generic substitution, and just look at this editorial which says that in their quest to gain early entry of new generic products prior to patent expirations, one of the strategies pursued is to incorporate different salts of an approved pharmaceutical ingredient in a brand company's marketed dosage and form, and trying to achieve bioequivalence. So let's look at it again. Are they truly interchangeable? New England Journal of Medicine looks at it and says that although legally correct, should you be using it? One beautiful editorial which says, generic medicines for you, but brand name medications for me and my family. At the end, I would conclude by quoting Shakyamuni Buddha, people who are vigilant do not die. People who are negligent and play indifferent are as if they are dead. Thank you very much for your kind attention.